So we're going to get started now. And Jeff is going to give a talk on GBS usage cases, examples from Maize. One of the things I want you all to think about here isn't so much necessarily about, oh, geez, we don't have exactly all the resources they do in Maize, which is, going to be the case for a lot of these examples. At least we don't have the resources now, right? What I'm hoping that you get out of this particular talk and some of the others that Jeff gives is, here are some possibilities. Here are ways of approaching problems. And as time goes on and the sequencing costs go down, we'll be, you may be in a situation right now where you don't have a reference genome to work with, or you may not have some kind of structure population or this or that. But this should give you some ideas on what is being done at the cutting edge of genetics research, and maybe, as time goes on, steer your things, say, well, geez, maybe we could do this if we did this kind of thing. So that's what I'm hoping you're going to get out of what Jeff has to say. Thanks, Jeff.
Okay, so I'm like a researcher in the Buckler Lab and project manager of the big Maize project that Ed has called Panzea. And that's just sort of part of my time. Most of the time I work on bioinformatics of GBS. Let's see. So I just want to cover today some potential applications of GBS, and from a Maize perspective mostly, with examples from Maize, but also make some comments that might be pertinent to other situations. So I'm going to cover these different things. I'll start off with the idea of GBS as a source of markers. And that's a bit of a strange thing to say because it's a marker system by itself, but you might want to develop markers from your GBS markers. One reason is that GBS can often be low coverage, so you'll have missing data. So you might be interested in a particular part of a chromosome, say where you have a candidate gene or a region of interest that's affecting a QTL peak is there or something like that, and you want to study that in more depth. Then you might want a marker in that region that that works on all the time with little missing data. So there you might want to convert your GBS marker to a PCR type marker. So that can be done. You can convert them to SNPs or PCR assays of indels. Converting them to SNPs is a bit tricky because the tags themselves are only, do we have any kind of pointer, I guess? Just my finger, okay. The tags, so these are the GBS tags. We trim them down to 64 bases, so that's not much room to design a SNP assay. But you could fish out the original reads that would be longer, and these days they're getting longer and longer. And if you also know that the tags are close to each other, you might use tags like that to make an assay, a SNP assay. And if you want to make PCR primers from adjacent tags, it really helps if you have a reference genome so you actually know that the tags are adjacent. So then you might design PCR primers off some tags like there and there or something like that, and hope that there are indels in there that you can score where you just PCR it up and score it on an agarose gel. And we've actually done that in some of our Maize Teosinte mapping projects where we're trying to map a QTL down to a gene, and we need markers to saturate or just to test for crossovers in that area. Okay. So yeah, I guess between the blue versus red here, that's trying to indicate that there's some kind of difference in that tag, so that could be a source of SNPs. And I think that's all I want to. Any questions or comments on that? Okay, or GBS could be used for phylogeny and kinship, and when we run GBS in Maize, we use APK1, we get a lot of missing data. So, but missing data is not that much of an issue for estimating pairwise genetic distance or kinship. And this is because you might have 100,000 markers overall, but a lot of missing data. So any two individuals, they might only share data at 10,000 markers, but that's still a whole lot of markers in common to share. So as long as your distance measure doesn't use missingness in the two different individuals that you're comparing, then it's fine. So in our tassel, genetic distance doesn't do that. So if both of them have n, it doesn't include that in the calculation. So you can get really good estimates of pairwise distance from a large random sample markers. So that's good. So this can work really well even in non-model organisms. For example, Fei Lu is a postdoc in our lab who works in switchgrass, and he made a nice phylogeny, and I'm hoping it's going to come up next here. Yes, it does. There's Fei. You'll notice Fei has terrific hair. He's really got great hair. He's like the Chinese Jimi Hendrix, I think. And he's been working in switchgrass, and he used GBS. Doesn't have a reference genome. He used this. He wrote a pipeline called Unique for doing this, which we'll talk about in a little more detail later. And when you look at this in detail at this phylogeny, you can see that different localities, the plants from those different localities are all grouping together. So clearly, the phylogenetics is working really well with GBS data. Yes.
How does he build that phylogeny? Does he concatenate all those markers across all his samples and generate a sort of alignment?
Well, you get a genotype file, and he just open it in Tassel and ask it to calculate genetic distance and make a dendrogram. And then you could, if you wanted, you can import the distance matrix out of Tassel and bring it into other programs and do other things with it. So, any other questions? Okay. And if you want to do a sort of PCA plot, a principal coordinates analysis is much better than principal components analysis, because principal coordinate analysis can withstand the missing data because the input to that is a distance matrix rather than every single genotype. So it's also known as multi-dimensional scaling, or MDS. They're pretty much the same thing. So yeah, so missing data doesn't bother you so much in principal coordinate analysis. But one thing, so traditional SNPs, like on a SNP chip or SNP assays that you design one at a time, they can be strongly affected by ascertainment bias. So ascertainment bias is when you use a small panel to discover the SNPs. So the SNPs have to be polymorphic in that small panel, and that can distort estimates of population genetic parameters such as kinship, diversity, demographic parameters, things like that. And so I have a really extreme example here from the Maize 55K SNP chip, also known as 50K SNP chip, same thing. So I'm just going to walk you through that. So we were interested in how much, how GBS compared to traditional SNPs for ascertainment bias. So we had a visiting scientist in our lab, Ram, who did this analysis. These are principal coordinate plots. And first he used SSRs, or microsatellites. This is the kind of picture we expect to see. So the green stuff there is tropical lines of maize, and we expect them to be pretty diverse, and they are here. And this one here is B73. That's the reference genome, the individual from which the reference genome was constructed. So that's like one of the most important maize lines. And another very important maize line is MO17. That's in a different heterotic group than B73. So when you cross them together, you get heterosis in the offspring. So it should be different, but not crazy different. So then if you look at, so that's what SSRs show you. Then if you look at the 55K or 50K, same thing, SNP chip, this is what it looks like. The tropical stuff is all crunched down to this little ball, and B73 and MO17 look really, really different. And this 55K chip, it has 13,000 SNPs that come from industry, a certain anonymous company. And when that company released those SNPs to this public resource, they had a policy where we're only going to release SNPs that are polymorphic between B73 and MO17. So that was their panel, just two individuals. So that creates this extreme ascertainment bias in a portion of the chip that was enough to affect the rest, even when you use the whole chip like that. So here the tropical stuff is crunched down into this tiny little ball, and B73 and MO17 are the two most divergent maize on the planet, basically. So ascertainment bias is not always that bad, but even if you have a panel of 10, it'll still be there. Whereas with GBS, Rob kept saying the discovery is done simultaneously with the genotyping, that's how he put it. And he kept hammering on that, so that gets rid of that ascertainment bias problem. So it gets much more like the situation we saw with the SSRs. The tropical are spread out, not quite as nice as the SSRs, but the B73 and MO17, they aren't crazy different. So any questions on ascertainment bias?
To make myself clear, is the ascertainment bias due to the small sample size?
The small sample size when you discover the SNPs. You decide, you go out and find your markers, and you do it just in 2 individuals or maybe 10, a small panel that you apply, or you sequence them or something and say, where are the polymorphisms in this small panel? And then you get that set of markers and apply them to more things.
Is there I don't even know how to say this, somewhat of a reciprocal effect where if you were to discover these SNPs on the whole population? I work with mussels, and we run a breeding program, but we work with a bottleneck or a sub-part of that.
Right.
What's the reverse effect if you find polymorphic SNPs in the world and then you're working with a constrained population?
I think that's when you're most free of bias, and that's the approach we take to GBS. We're kind of doing these talks out of the usual order. We normally go through the pipeline first. So in the pipeline, we have a discovery pipeline and then a production pipeline. And we do these periodic discoveries in Maize where we use all the samples we have of Maize. We throw everything in and we discover the SNPs. So that's what we advocate, and that'll get the least amount of ascertainment bias. So yeah.
So that's, and earlier today I was talking about this as a species-wide genotyping tool, and just throwing everything in again and again, and getting as much information on the species level as you can, right?
Of course, you can't. I'm so tired. I'm sorry.
A lot of those want to be uninformative and restricted.
I guess you
Yeah.
can produce variation where it doesn't exist.
But say you want to.
If you have a mapping population, there are only a certain number of markers that are going to be segregating in that population. But some people will say, well, I want more markers, so I'm gonna change the filtering parameters so I get more SNPs for my mapping population, right? But that's introducing now error.
But say you want to measure how restricted was that population, then you want to have that so you can measure it, right? So
how wide was my cross compared to the diversity that I see in my species?
Okay, so I'm going to say a bit about linkage mapping, but in Maize it's not something we need to do. So in Maize we're lucky, we have a great reference genome. We also have a lot of resources. We have a lot of inbred lines, and we have a lot of crosses between those lines and real populations that we can work with. And those are really great to make GBS work. So in Maize we use the reference genome to order the markers, and as I'll talk a bit later, sometimes it's wrong, but for the most part the reference genome is really good, especially for B73 itself. So if you don't have a reference genome, then you'll need to order the markers in a biparental cross for linkage mapping, and you'll have to do the order from scratch or de novo and use software to do that. But if you use something like APK1, you'll have too many markers that'll swamp the software. It can't do it. So the obvious thing to do is just filter for the markers with higher coverage and get a smaller set of the highest coverage markers that the software can handle. Or you could use a 6-base cutter or combination of enzymes to get fewer markers with higher coverage to start with, so you're not throwing away as much data. And I'm told that since I work in Maize, I don't use these softwares, but I'm told that JoinMap can use about 3,000 markers, but there's even better software out there, newer stuff. MSTmap can do at least 10,000 markers, and QTI, ICI mapping can do about 9,000 markers, and there might be others out there as well.
Jeff, I can just say the latest version of JoinMap does a minimum of 50,000 markers, so that you can order at least 1,000 markers per linkage group.
Per linkage group, right.
So
How fast is it?
He didn't say days. I
So-
when I was talking to him, Peg is saying I think it's sort of like, it could be sort of like a moving line to maybe a day or so.
So Andrew, just so that this gets recorded as we're going, you said that you could use up to 50,000?
He said minimum 50,000, I think.
Minimum?
Maximum.
Maximum.
Maximum up to about 50,000.
Up to
Right.
about 50,000 with the latest version of Join Map.
About 1,000 per linkage group, is that
He
right?
said that
Yeah.
you can get sort of effective ordering of about 1,000 most.
Right, okay. Okay, so I'll probably need to update this slide. Thank you. Yeah. So when we in Maize we have this low coverage situation with APK1, and we have real populations, but they're not completely homozygous, so they have some heterozygous segments in them. So we need to be able to call those things and fill in the missing data. So Peter Bradbury in our lab developed a HMM, hidden Markov model for doing this. And it basically tries to figure out the probability of the actual genotype given the state that you observe. And tries to figure out what the heterozygous segments are and what are mistakes in there. So this will be a little clearer with this example here. So this slide shows 3 different rills on it: a low heterozygosity rill, a medium heterozygosity rill, and a high heterozygosity rill. So they're not complete inbred rills; they still have some heterozygosity in them. So let's just look at the medium one and say this segment along here. So the top graph in brown is the actual observed data coming out of GBS with low coverage. And then the one in blue is after applying the HMM model and figuring out what the segment is. So the bottom here is the B73, which is the reference allele, and then the top is non-B73 homozygote. So those are both homozygotes, and the middle is a heterozygous call. So this is just the raw data, and this is a heterozygous segment. So if you have low coverage, sometimes you've only sampled one of the 2 alleles. And if it's heterozygous segment, you're sampling them at random, basically. So the calls are flip-flopping between heads and tails as you go along that segment, and sometimes you actually get 2 or more reads on that genotype, and you can call a het. So this is obvious signature of heterozygosity, heterozygous segment, and so Peter's algorithm can recognize that and call that whole thing heterozygous, and then just fill in between any 2 points that agree on either side. The missing data in between just fills them in to what the flanks are called. So that's basically how his thing works. If I go, and it does that by, it's a little bit magical, which means I don't fully understand it enough to explain it in detail, but it walks through this graph of all possible states here. So this is just showing 4 SNPs here in a row along the chromosome, and this thing, to represent an entire chromosome, would go to the other side of town, basically. But this could walk through all the possible actual genotypes that are there, and this is the path that you could walk. And without having to walk it over and over again, it can walk through a very small number of times and figure it out what the best state is. So the distance between these nodes is the probability, so it finds the maximum length walk through this thing very efficiently. So Peter coded that up, and it works great. So especially in biparental rails, it works fabulous. Okay, but before he had done that, I did some work in a Teosinte W22 cross. So Teosinte is the wild progenitor of Maize, and W22 is an inbred line that grows pretty well in Wisconsin. And John Dobley in Wisconsin likes to study the genetic effects of domestication in maize and what are the important genes and things like that. So he built this population to study that, and we use GBS to genotype it. So the population is a BC2S3 back-crossed twice. So W22 is a recurrent parent, and then it was self 3 times, so there's still a lot of heterozygosity in it. So the population had 868 rills, and John already had a framework map with 492 SNPs. I think they were from the company Syngenta. And so he could provide me with that framework map, and then we ran GBS on it, and I ended up with just over 50,000 biallelic GBS markers from that, and I used the framework map of SNPs to help sort of seed pulling out the good markers out of the GBS. And this table just shows running it. We ran these samples on different versions of the Illumina machine, and we get a lot higher depth with the newer HiSeq. So I used my own Peter's algorithm. The HMM wasn't available at that time, so I came up with my own algorithm to sort of do the same thing. And this shows what the genotypes for a whole chromosome looks like across these 800 RILs. So think of it as an Excel spreadsheet or something with colors. Or how about an OCC tab delimited file with colors? Okay. It actually is from a tab delimited file. And then so Peter had, so if you use Tassel at all, and you will later on, when you look at the genotypes in Tassel, you can only look at a little bit of it at a time, and you have to sort of move around it. You can't see the whole big picture. And I want to see, does my algorithm even work? Does it look like it makes sense or not? So Peter had also written an R script to take the genotype file and make these plots. So I took his R script, and of course I have to fiddle with it and change the colors and make them uglier and stuff like that. So, so that's what I did, and so I came up with this. So each row on this thing is a RIL, so there's 868 rows, and each column is a marker, which for me was a tag. I used the tag itself as a marker. So I either had the tag can be a W22 allele, so that's in gold, or a Teosinte allele, that's in blue, or you could have a heterozygous segment. So one thing you can see, and this is all of chromosome 9, and you can see right away, it's quite obvious that in the paracentromeric region, there's a lot less crossing over. We know that in Maize, the crossing over recombination is suppressed there. It's a lot harder to see, but if you look really carefully, a lot of the white parts are the missing data where I wasn't sure where exactly the crossovers were. So I left them, I was very conservative about that and left them white. And it tends to be harder to know when you're going from a heterozygous to homozygous transition, just where does the coin flipping stop and where do you get run. So those white spaces tend to be a little longer. Yeah, so how do you look at this data? It's nice if you can, like Rob said, learn to code something, and then you can just open it up in R and make a little plot, which is, this is a PNG plot, I think. Okay, so reproducibility. So when John was very skeptical about GBS, and I think this little data set convinced him, but what he did was 60 of his RILs, he gave me 2 samples of them. One was from a leaf tissue and one was from seedlings, but from the same RIL. And he wanted to see that we get the same results. So, and in the initial plot, I just combined those 2 together and used them as one individual. But here I split them out. So, these are all in groups of 3. So we got groups of 3 across the 60 RILs. So here's a group of 3 here. So one of them is the DNA from seedlings, the middle one. The bottom one is the DNA from leaf tissue, and the top one is the combined data. And this just shows that in general you get really good reproducible results, but it's not perfect, but it's pretty good. And I think when you get some differences, it's probably because maybe the lower coverage, then I'm not as sure how to call things. It might be more of that than the lack of reproducibility of the GBS assay itself. Okay, so this map that I got, they then went and used it in a collaborative study where folks working in sorghum had looked at a gene that had been cloned in, they basically positionally cloned a gene in sorghum that also had been cloned in rice. It's called the shattering 1 gene. It controls seed shattering. So when you domesticate a crop way back in the day, you want to find a mutant where all your corn kernels don't just fall all over the ground, so you don't have to pick them up. They stay on the cob. And same thing for rice, same thing for sorghum. So the very same mutations are not the same mutation, but mutations in the same gene, sort of a knockout of the same gene, has been selected for in parallel in different crops. Okay, so in maize they found, so they knew what this ortholog was, and then they took this map that I developed and they took this, they scored in those individuals seed shattering, and they got 2 QTL under which the maize orthologs of the shattering 1 gene was found. So it showed that the very same genes were selected for during the domestication of maize. Any questions or comments there? So in our project, at least the last round of our project, we were very ambitious and wanted to go out and find map a lot of QTL. The reason we want to do that is we'd like to do a lot of GWAS, genome-wide association studies. And when you do that, you get a result, and you see, oh, there's a peak there. There's some SNPs with a really low p-value. You know, they're associated with the trait, but we need some positive controls. You know, we need to know we should be hitting this. So, and so we wanted to find map things, first of all, to show it could be done, and second of all, to have positive controls for GWAS. So in order to do that, first you go out and get a QTL like I just showed you, but under that QTL there can be, it's just a sort of chunk of the chromosome that you know is associated with the trait, but you don't know which gene in there, and there could be hundreds of genes in there. So if you really want to go down and figure out which gene, or if there is only one gene in there, sometimes it's multiple genes, that affect the trait, then you're going to have to saturate that interval with markers. So GBS is a really good source of markers that you can quickly do that with. But you also need to collect recombinants in the interval. So you might start with taking a couple of GBS markers, convert them to a PCR assay, and just use those flanking your region, and then start generating recombinants and use those markers to say, are there recombinants in here? I don't know where they are yet, but I just want to collect a bunch of plants with recombinants. So if you're lucky enough to be able to generate near isogenic lines, it's really going to help. So you get a near isogenic line where that chunk is different, but everything else is the same. Then you make that heterozygous, and then you start getting recombinations. You just grow out tons of plants, genotype the flanking markers, look for recombinants, and collect them. It also mendelizes. It can be a somewhat quantitative trait, but if they're nils they'll mendelize the trait, so it'll be easier to score this group versus that group qualitatively. And of course, you need to have a good reference genome for this. If you don't try it at home. So the basic idea is, this is kind of schematic, but you really just want to, if this is the actual gene that's causing this QTL, you want to have a crossover on either side of it to be able to sort of prove that that's the gene. So again, in John Dobley's lab, they were trying to find map some domestication QTL, and they have been pretty successful on some of them. This one's unpublished right now, where we use GBS data, so I'm just calling, and there was 2 traits they were after on some chromosome, some mystery chromosome, but the traits are trait Y, trait Z. I've changed the names to protect the innocent, basically. So I could share this result with you. It doesn't really matter what the chromosome is or what the traits. But so this student in Dobley's lab was interested in, first of all, trait Y, but unfortunately trait Y, when they do the QTL analysis, it falls into this paracentric region where there's not much recombination. So he tried hard, but he wasn't able to get a lot of recombinants in that region. Where the trait Z, or Z, it's in a better place, a lot more recombination. He did a lot of work to collect all these recombinants, and he started with just flanking markers to say, oh, there's a recombination in there, keep it. And then we ran GBS, and we now say, well, where is the recombination in there? And we get the idea where it is, and then he can grow out these plants, the ones that sort of define different bins, and grow them out and measure the thing and try to really narrow down where where that's what the gene is underlying that trait, and that's ongoing work right now. Okay. Any questions there? Is that clear? Okay, so some comments on genomic selection and GWAS. So people familiar what genomic selection is? Raise your hands if you know. Well, genomic selection is when you try to use markers to accelerate breeding. So you kind of do a regular breeding, but you not only do your phenotype, your genotype, and then you try to make a model between your genotype to predict the phenotype, and then apply that model to speed up the selection cycle. So you could just grow up lots of plants and have them as seedlings or even seed and get DNA from them, and then just use that model to say, oh, these are the ones to keep out of, and do like a high selection intensity and faster cycling. Okay, so that's what it is, and so you could. That's one possible application of GBS markers, one that we're very interested in. So it's called genomic selection. Sometimes it's called genomic prediction. And even if you have a lot of missing data, you might be able to get away with very basic imputation approaches. So you could have your non-missing data, and you could code it numerically so that one homozygote is called a 0, the other homozygote is called a 1, and the heterozygotes are called 0.5. And then from that data that's non-missing stuff, you could calculate a mean genotype and then put that in for all the missing data. That would make all the missing data kind of non-informative. So that's one way approach to like imputation, just to do genomic selection. And Jason Pfeiffer was a grad student in our lab, and he was seeing if he could predict in 2,800 diverse Maize lines, if he could use the genotype to predict the phenotype, to predict the height, basically. So he did something called cross-fold validation, where you use like 90% of the data as a training set, and then see if you can predict on the other 10%, and then do that 10 times. So he did that for these lines, and he could predict really well height, but this isn't totally a proof of concept of genomic prediction or genomic selection. Does anybody have any idea why? When you do GWAS, it's really important to control for population structure because it can give you false positives. So when you do genomic prediction and you don't take account for population structure, then the markers that can just be telling you what population it came from. And tropical lines, when you grow them up north, they tend to grow taller than temperate lines. So he's probably just predicting which population they came from more than, and that's not the same as doing genomic selection. That's like almost population replacement instead of breeding, right? So it's not totally a proof of concept yet, but genomic selection and genomic prediction is a really, current area where we're really trying to use markers to help feed the world, I guess. So accelerate breeding. So then GWAS is genome-wide association analysis. You're trying to figure out which actual find SNPs that they're not actually the causative polymorphism for some trait, but they're in high enough LD with that polymorphism that they sort of track it. So that's what you're doing with GWAS. So you're taking some trait and you're scoring it a bunch of samples in a common garden experiment, and then you're throwing many markers as you can at that. So that's why in Maize we really want to have a lot of markers. The trade-off between a lot of markers with low coverage versus fewer markers with high coverage, we have to go a lot of markers with low coverage. Otherwise we'd have no hope of making GWAS work. So that's why we use APK1 in all our Maize stuff in the Buckler lab. So we now have 50,000 Maize samples all scored with APK1, and we're working towards 90,000. So if you try to do GWAS and you have this missing data, and you need to impute the data, otherwise you can't do the GWAS. But if the imputation is wrong, it could cause spurious results. You might get false positives. Also in Maize, there's a lot of structural variation in the genome. So a piece of DNA that's in one line, like B73, might not be there on that same spot on the chromosome. It might be just missing in Mo17, for example. So when you're imputing, you'd want to try to take into account these missing regions, so it's a tricky area. So we've been working on that pretty actively for the last couple of years, and I think we're happy. We've kind of decided we've gotten as far as we can, and I'll be talking more about that tomorrow, I think, our current approach to imputation. So you need to do something more than just this basic genomic selection approach of using the mean. So using not our most current imputation in inbreds, but our one from a year or so ago, Cinta has done GWAS in about 1,600 diverse inbreds from the USDA Ames collection, which is like worldwide inbreds. And the first thing she does, let's try a really simple trait where we know what the gene is. This is like a positive control. So for kernel color, we know that the Y1 gene controls kernel color, and when she does a GWAS and uses the kernel color for these inbreds, uses the GBS markers, she gets a direct hit in that gene. So if you got a Mendelian trait, it can work really great, and say you didn't know what the gene was and you wanted to know, it can be amazing. So, and it can even work for more complex traits that are controlled by more genes, like flowering time. So this shows that Cinta hits a number of flowering time genes, and a lot of these are QTL we've already known about from previous studies. But the largest one is up on chromosome 10, and she just hit it with one SNP, so she just got really lucky that she had that SNP in her data set. And so these are called Manhattan plots because they look like skyscrapers and things, and that's like a helicopter flying over Manhattan. So she's lucky she got that helicopter, and that kind of illustrates that you really need a lot of markers to do GWAS. When linkage disequilibrium decays really quickly along the chromosome, which it does for unrelated Maize lines. So she had 660,000 SNPs here, but she almost missed the most important QTL. So if you want to do GWAS, you need lots of markers.
How would you check that that's not an artifact or an outlier or something?
Well, we already knew from previous, that's one of our positive controls, you know, where it's been studied in other populations, and it's even been fine mapped, this one, down to which gene it is. So we already knew that that's a very important gene for flowering time. But if you don't, then you don't always know. So that's the trouble. That's why we wanted to fine map things and have more positive controls. But that's so people will, okay, let's look at what we hit. Now let's go look at what the genes are. And if you go do that, like that how do you say that? Not a priori, but post hoc or afterwards, you go in and say, well, what did I hit? And these are the genes, and oh yeah, that makes sense. You can always look at what a gene does and make a story. So you really have to pick your list of genes. To do it right, you have to pick your list of candidate genes before you do the analysis and say, these are the ones I think might be important. When I do this analysis, do I enrich for this list? Do I hit more of them than I would expect by chance? That's a kind of one way to go at it. But it's true. That's one problem with this area. You don't really know. That's one beauty, though, of genomic prediction and genomic selection is you don't necessarily have to know. Like you can still use the information in breeding. And the difference between genomic selection and just marker-assisted selection, marker-assisted selection is when you get some significant QTL and you try to use the markers linked to those to try to just like enrich for those alleles, those good alleles at just a few loci. In genomic selection, genome prediction, you use all the markers across the whole genome and just throw everything in. And that way, even the stuff that doesn't pass the significance threshold, you can still use that. So you don't necessarily have to know depending on what you're doing. But, yeah. But to get a paper in a high journal, you probably want to know. So, yeah.
To rationalize it well.
So our second questions. So if you think that one is a true positive.
Yes.
Is the truth. The second one is the huge differences with the first one.
Like this one?
The orders.
This one here?
Oh. The first one, the second one. I think
Oh, the drop off in the p-value, you mean?
There's 10,000 differences.
Oh, yeah,
So
because
how-
this is a log
yeah.
scale. Yeah, yeah.
And
And you
generalize the criteria is a positive call.
sorry,
These are
can
real markers.
yeah, we tend to, I'm not showing it, but we usually do a QQ plot here too. So if you control for population structure well, then it'll follow the line, but then on the most significant stuff, it'll really jump up. So these ones will be way off the QQ line, which would be what you expect if there was no actual signal there at all. So that can help you trust it more. But you probably, one way you could do it is do an independent experiment, another one, so maybe in another set. So, and that's what this is in a sense. We just first discovered these QTL in the NAM population, which is our big Maize population of 25 families across the B73. That's like a joint QTL mapping population. So we discovered it there, and here this is a totally independent set of unrelated inbreds. She hits it again. So we believe it. We believe that gene. But you don't always know. You're more confident when the p-value is lower. So yeah. Okay. Oh, and this GWAS plot was done by a tool developed by Alex Lipka and Jiwoo Zhang in our lab. The tool's called GAPIT. Okay, so I've been talking I've already said a bit about NAM. So in NAM, GWAS imputation is less of an issue. And so what is NAM? So NAM is the nested association mapping population. It's a collection of 25 diverse lines. That's what DL stands for. Diverse inbred lines. They're sort of picked with SSRs to really get as much diversity as you can, but also include some of the most important Maize lines. And they're crossed to B73, which is that reference genome. You take the F1, and then within each family, you develop rills by selfing with single seed descent. And in each little family, there's 200 rills. So they're really just a set of 25 biparental rill families, but they all have a common female parent, B73. So it kind of gives this unified population. So we built this, I say the we in the royal sense. Our project, the Panzea project, built this population for what we call NAM GWAS. And our approach there was for B73, it's the reference genome, so we know the entire genotype of the thing. For these ones, we used next generation sequencing with random shearing, whole genome sequencing, and then we called SNPs against the B73 reference. So we call that the HapMap project, the Maize HapMap project.
Can I interrupt just for a second? So
Sure.
that's where I got my start in next-gen sequencing.
That's right.
That was my first work in next-gen sequencing, was figuring out how to make reduced representation libraries in the very beginning of Illumina stuff. And it was that experience which helped inform me as I developed GBS, and I think it's kind of useful to share that sometimes.
Right, yeah, so it spun off into GBS later.
Yeah.
So he cut his teeth on HapMap 1, Maize HapMap 1, and then we did a Maize HapMap 2, and in Maize HapMap 2 we ended up with 55 million SNPs. I think about 26 million of them are polymorphic in this set of 25 or 26 individuals here. So we've basically painted the chromosomes of the founders of this population, and then we can use GBS in all the RILs to figure out where the crossovers are and project down these dense genotypes from the founders. And that's a general strategy that you could use in lots of different things if you have like a set of founders or even a set of reference haplotypes. Like in humans, they have something they call the 1,000 Genomes Project, where they've done whole genome sequencing on 1,000 genomes, and then they do SNP chip genotyping on hundreds of thousands of humans, and then they try to use those lower density SNPs to project the haplotypes of the thousands on so they can get dense genotypes for everybody, and then they do disease association studies like GWAS of disease. So this is how we did it in NAM, and so initially we only had 1,100 SNPs, like for from sequenome, I think, in the NAM rills. But even that worked pretty well. So we got this nice paper in Nature Genetics, which got totally messed up on the slide here. It doesn't look anything like the original, but the key thing here is that it hit the ligulus 1 and ligulus 2 genes for the trait leaf angle, okay? And leaf angle is a really important trait in domestication and continued improvement in maize, because as you pack more maize plants together and get more productivity of land, the leaf angle had to adapt to that, right? So a ligule is sort of this membranous bit of tissue here right between the sheath and the blade of the leaf. So it's sitting right there at the hinge. So it makes sense that a gene called ligulus that affects the development of the ligule sitting right at the hinge would probably affect leaf angle. So hitting those two genes made a lot of biological sense. So that was like one of the first proofs of the NAM GWAS concept. So we're using GBS, as I was sort of saying, to improve upon having these 1100 SNPs. Now we have many more GBS SNPs. So if this were two of the NAM parents, the B73 and one of the other 25 diverse lines, and this is one of the rills with the 1100 SNPs, with GBS it gives us a higher density so we can pin down that the recombination, where the recombination occurs with more accuracy. This should give us more accurate and precise GWAS within NAM. And Peter has shown that indeed it does. We get higher accuracy, higher precision mapping of QTL in there. And we're also studying, using GBS to study recombination rates in the NAM population. So again, and this is a plot of recombination rates across all the NAM families for 10 chromosomes, all 10 chromosomes in maize. And again, you see recombination is really low in the paracentromeric regions. Recombination is high in the telomere. We're interested in things like looking at the pairwise parents and looking at chromosomal segments and saying how different are the segments, how related is the recombination rate to differences between the individuals, like structural variation and things. We're also interested in how recombination relates to heterosis. We tend to see that residual heterozygosity in NAM is higher in the paracentric region, and we think that's because of the low recombination there. When there's a couple of bad gene, or there's a bad gene here, it can't be recombined away as easily because the recombination rate is low, so you can't get rid of it. So you need to be crossed to another individual that has a good gene there, and that's the basis of heterosis. So those two individuals will sort of reinforce each other, complement each other at multiple genes across this paracentromeric region. So we think that's a basis of heterosis in maize, or at least that's Ed's favorite theory. Okay, so the last part of the talk is talking about some work I did with GBS to improve the reference genome. So it's a good reference genome, but they always go through versions, just like software, right? So now we have V3, but they started with V1. So there's a few different things I looked at, 4 different things I addressed using GBS markers, but I'm just going to talk about 2 of them today. But I'll just walk through what they are at least. So first of all, the B73 reference genome is really good for B73, but not necessarily good for other maize lines. That's because maize lines are so different from each other. Between the genes, they may look completely different. So that's what we call structural variation. But even for B73, when they put the genome together, they assembled it, and they didn't necessarily have the parts list and assembly instructions. They figured it out. Sometimes they made mistakes and they put regions in the wrong place. And also there's some parts, have you ever gotten a big complicated thing to build and you build it up and then at the end you have some pieces left over and you're going like, oh, where did these go? Well, they didn't know, so they stuck them all in something they called chromosome 0. So in RefGen 1 there was 30, then they were able to place 13 of those in RefGen 2, and the GBS data supported those placements. And also some regions of the genome are supposed to be there, but they're missing. So there's actual B73 sequence that is not in the reference because they missed it. You know? So it's not even on chromosome 0. So I'm just going to address the first two so I don't prattle on too much longer here. And okay, so to talk about how to do that, we can map the tags as individual alleles and ignore. We don't have to, we can just focus on the one tag from either parent in the biparental cross and ignore the other tag and just score it as a presence-absence marker, as long as it's polymorphic between the parents. So this one's blue and that one's red, so that shows that they're polymorphic, so I can map that tag and ignore the other one as a presence-absence allele. And, or this one is missing, so I can map that tag. It doesn't matter, that one's not there. So how do I do that? I use a binomial. So I have the 55K SNP chip data for the IBM mapping population, B73 cross Mo17. As I showed you, that's a great chip for that family, right? So this is like a representation of some of the data along part of the chromosome. And this is external framework map, right, of SNP chip data. And these are the genotypes. Blue is B73, red's Mo17. There's some heterozygosity, so yellow is heterozygote, okay? So then I want to ask, well, does a GBS allele map to one of those places? Like, for example, does it map right here? So let's just split this thing open and ask that. And so now I'm following the segregation of this tag. That it's a blue tag, so it's a B73 tag, so it matches the reference genome, for example. So I'm following the segregation. So I'm going to test that across this whole entire framework map, across every chromosome. But this one actually happens to map here, because every time you see blue, it lines up with blue. Blue, 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 blue. The blue tag points to a blue SNP every time. So that's like flipping a coin and getting 10 heads in a row, right? So that's what the binomial test for linkage. So the beauty of this is we totally ignore the absence of the tag, which could mean where we don't have the data for the tag, it's either could be a Mo17 tag or it could be missing data and we don't know. So we just use the presence of the tag. So our sample size is 10 here, how many times we observed the tag in the RILs. So to get that, we of course did GBS on all the RILs. So that's how we can kind of map the tag through the genetic segregation. So we can do it for B73 tags, and we can test how accurate this is, and that I didn't mess up the code anywhere. And we can do it for Mo17 tags as well, and see where they go. So when you have missing data, you only have so much resolution. So actually, all these SNPs here are equally good placement for this GBS tag. They all tie. Okay, so I can do that with different p-value cutoffs. So for my next slide, I focus on this higher level p-value cutoff, and I have about 74,000 B73 tags and 67,000 Mo17 tags mapped like that. So I plotted for just things that align to chromosome 1. Okay, so here's B73 tags that align to chromosome 1. When you do an alignment, a sequence alignment, they align to chromosome 1. But then asking, when you do the genetics, like I just showed you how I did the segregation test, and say, where do they map to genetically, okay? So most of the time, they map to chromosome 1, right? Genetically as well as physically, which is good. So there's only 0.4% of the tags that genetically map to a different chromosome than the one that they align to. So that's could be sampling error or little mistakes in the reference genome. So that looks great. B73 reference genome is really good for B73. But you can turn around and map those Mo17 tags. So you can map the red ones. So we can take the ones that align to chromosome 1. This is where they align on chromosome 1. And then we can ask, where do they map to genetically? And a lot more of them, more than 9%, will genetically map to a different chromosome than the one that they align to. And that's because there's so much structural variation in maize. Any two maize plants only share about half their genome. It's usually the genes, and the stuff in between the genes is changing. It's got all this transposon variation. Sometimes the genes are coming and going even. So any questions on that part? Okay, and some chunks of the genome are in the wrong place. So here's a sort of table ordering those chunks by their size. So there's part of chromosome 10 that is placed to around 139 on chromosome 10 by physical methods in the reference genome. But if I genetically map, there's a section of 49 tags in there that actually genetically map to chromosome 2. So they got it wrong. And the same for, there's a bit of chromosome 9 that at one end of chromosome 9 that should be at the other end of chromosome 9. And then they get sort of smaller as we go along. So there's a few mistakes in there, but not many. So these different mistakes and things, the GBS data showing where things should be fixed has been incorporated into version 3 of the Maize reference genome, which is out now. So I think, yeah, that's all I wanted to cover today. Is there any more questions or comments on any of this?
So do you guys, can you see some potential applications in your own work of the things that Jeff has described in Maize? Yes, I'm seeing some hefty nods. Yep, okay.
Maybe you can think about it for a few days too.
Yeah.
And get some good sleep, and so sleep on it.
I think we're biased about wanting some good sleep at the moment. So, well, thanks for that. Thanks for that, Jeff. I think it was a really nice illustration of what you've been doing in Maize. I think you guys have also seen from Jeff's talk, a number of different tools that were developed within the Maize community to do different kinds of jobs. And I think that's useful for us to think about, again, as you'll be hearing me talk over the next day and a half or so about community building and working together. We can get a lot more done across lab groups than just within one. I mean, these resources that Jeff spoke about are really, didn't just come out of the Buckler lab by any means.
No, they can't. A lot of it's Panzea project plus collaborators as well.
Plus collaborators on top of it. So that shows you the power of collaboration, especially over the long run.